let's see, physical locations of genes on chromosomes and how you know what the map distance is between them. Okay. So we have genes A, B, C, D, E. And I suppose we could make this a dihybrid. Actually, I guess that would be a pentahybrid. It doesn't matter. I'm not going to do anything crazy, don't worry.
Okay.
Oh, except I used C. My little C's look like my big C's. Go figure. All right. Now, we've got two things to consider. We've got independent assortment, what happens to genes that are on this chromosome. Let's number these. So this is chromosome 1, that's chromosome 2. So, will we ever get any big A's into one gamete with little b, little c, little d, little e? So will that chromosome there ever wind up in a gamete, sperm or an egg, but with that chromosome? So, if we do, and I'll just track one gene at this point. So if we do synthesis, then each of those 2 DNA molecules becomes 4. So you've got the tetrad of chromosome 1. There are 2 big A's, 2 small A's. And on the other tetrad, I'll just look at gene E. So those are all in a cell. And if that cell is going to divide, this is going to be the first division. This is just before the first division of meiosis 1. It's going to divide, I'm just saying, it's going to divide right there. So how many A genes have to go on the left side, and how many A's have to go on the right? 2 and 2. So you're free to tell me which way you think they're going to orient. It doesn't matter at this point. You can have the capital A's on the left and the lowercase on the right, or vice versa. So we can put the lowercase, I'm just going to arbitrarily choose to do it that way. And now I'm not even drawing the lines representing the chromosomes, I'm just writing down the haplotypes. So we got little a's on the left, big A's on the right. And this always happens. So the capital letters, the 2 versions of the same allele, the 2 copies of the same allele, always go together at the first division of meiosis.
Oh.
So you'll never get a big A and a little a on one side and a big A and a little a on the other side. So I'm going to draw another cell undergoing the exact same process. So a second meiosis happening from the same individual. So we'll leave the little a's the same. So they're going to segregate like that. So now how do I write the e's? Half of the time, the big E is going to be on the left side. And half the time, big E is going to be on the right side. Because these are separate sets of chromosomes, so how they orient themselves in 3-dimensional space inside the nucleus is arbitrary. They're just rotating freely in space when they line up across where the cell's going to divide. So half of the time, then, you get little a's with big E's and big A's with little e's, and the other half of the time you get little a's with little e's and big A's with big E's. Come in if you like. So after the second division, those would be the products of the first division. Yeah. After cell division. And after the second division, you'd get a little a with a big E. You'd get a big A with a little e. And equally frequently, you'd have the little a's having lined up on the same side as the little e's, and the big A's with the big E's. So from one meiosis, this is not meiosis 1, this is one round of meiosis, if the genes line up with the lowercase on one side and the uppercase on the other, you get an equal ratio, a 1-to-1 ratio of those two gamete types. But by chance, the second time this organism had a meiosis occur, you got the opposite arrangement of little a's with big E's and big A's with little e's. And that generates a frequency of 50% of each of those two types of gametes. And if you imagine meiosis happening 1,000,000 times, which is an overstatement for most organisms, but so imagine that after 1,000,000 meioses, you get 500,000 by chance with that arrangement, with the big A's and the little A's and the little E's together, and the big A's and the big E's together. And over large numbers of meioses, then the other 500,000 times this orientation occurred. So ultimately, you do wind up with a 1-to-1-to-1-to-1 ratio, 25% of each of those four types. And that's independent assortment. When genes are on separate chromosomes, half are recombinant, look like recombinants, and half look like parental types. And I didn't define which was parental and which was recombinant, and that's fine. So the point is that if genes are on separate chromosomes, you'll get 50% recombination. Figure out which ones are recombinants and which ones are parentals, you'd have to know something about the cross that made this individual. You'd have to know what the P0 parents looked like. So for example, actually we don't even have to write the chromosomes, we just write the letters. So let's say it was A's, two capital A's on one chromosome in the P0 generation. This is an F1 generation. And little e's. And that was crossed by a parent who had the opposite genotype if you're going to make an F1 dihybrid. So the other parent would have had to have little a's and big E's. So parent on the left has a gamete that's big A, little e. Has to be, because they've only got big A's and little e's. And the same is true over here. Their gametes have to have little a's and big E's. So that A came from there, that little a, that little e contributed there, that little a came, that's where that came from, and that's where that big E chromosome came from. So now we know the parental haplotypes. And only because I've told you this. By telling you the genotypes of those parents, you can figure out what their gametes look like in terms of genotype. So now we know the parental types, we could go back down. So it's capital letter with lowercase, or the parental types. So we know that big A with little e is a parental type, big A with little e is a parental type, and these are the recombinant types. And half of them are, 2 out of the 4 are recombinant types, and 2 out of 4 are parental types. 50% recombinant map distance. So if 2 out of 4 are recombinant, those 2, we're looking at 4 total gametes, then what's the recombination fraction? 2/4. Yeah, 2/4, 50.5. RF, recombination fraction, or multiply that by 100, 50 centimorgans. So when genes are on separate chromosomes, they line up separately in both of these fashions equally frequently during meiosis. So half of the gametes look like they're recombinants and half look like they're the parental types. And so that's why, in this case, for example, we can define that the A and the E genes are 50 centimorgans apart, and the A and the D gene, and the A and the C gene, and the A and the B gene, by virtue of them just being on separate chromosomes. Independent assortment will guarantee that all of those are 50 centimorgans. They act like they're unlinked because they are unlinked. They independently assort. And then the second part of the question was about the genes that are on the same chromosome. So how do you know the relative genetic distances between B, C, D, and E? And the only thing I can say about this is, well, the only thing I should say about this is, it's all relative. So we know that from the top to the bottom of the chromosome, genes would act like they're unlinked, because the recombination event we talked about in the review session, a recombination event has to happen between genes that are at the very physical opposite ends of a chromosome. And so in the study guide, the reason I concluded that the R and the T genes were not 50 centimorgans exactly, but they were less than 50 centimorgans, is like I've drawn here. None of these genes, no pair of these genes are at the actual opposite ends of the chromosome. E, I meant to draw that way. So E really is at the bottom of the chromosome. But B is not drawn at the absolute other end. So there's a chance, most of the time, recombination events will land in between B and E. If I just close my eyes and I just draw recombination events happening at random, most of the time recombination will happen between B and E. And the more time recombination happens between two genes, the closer and closer the recombination fraction gets to 50%. But occasionally, rarely even, if that B gene isn't at the top of the chromosome, it's just close, there will be a recombination event that happens there. And when recombination happens here, you get no recombinant types produced, which means the recombination fraction here, so if we tally these, there are 1, 2, 3, 4, 5 recombination events that cause recombinance and one that does not. So recombination fraction is not going to be equal to 0.5 because there was one recombination event that didn't make big B with little e or little b with big E. So that means that with the addition of that recombination event, recombination fraction is less than 0.5. So the only thing you could conclude is that they're less than 50 centimorgans apart. Relative to B and E here, C and D must be even less than the distance from B to E. And I would not ask you to do something like rank all of the pairwise genetic distances, because there's no way to do that. We've got two genes, A and B. So you've got a dihybrid.
Okay.
So in this case, you write all of the letters that are part of one molecule of DNA next to each other with nothing separating them.
Okay.
That's
when they're linked.
That's when they're linked, right?
Okay.
So this is linked, and that constitutes a haplotype. So that's what I refer to when I talk about a haplotype. It's the genotype of, here is a diploid. You've got two chromosomes, two copies of each gene, diploid. So one copy of a chromosome is half that, or haploid. So a haplotype is basically the genotype of one chromosome, one physical piece of DNA.
Okay.
If we were going to write the genotype, that's two haplotypes.
Okay.
So in that case, it's big A, big B over little a, little b.
Okay, that makes sense.
If they're on separate chromosomes, you still have the same genotype.
Oh, wait, the actual picture, the real picture? Oh, yeah. That was also in today's. You went over that.
But because these genes are on separate chromosomes, so you still have a big A, a big B, a little b. This is where you write haplotypes with slashes, but we know that these are on separate chromosomes. That's where you put the semicolon and then the other genotype.
So are these on different chromosomes as well? Or
so yeah, exactly. When I say chromosome, I technically mean pair of homologs.
Okay.
So these are the same chromosome in that, for example, say this is human chromosome 1.
Yeah.
Your diploid, you've got two copies or two homologs,
Okay.
and then this is a second pair.
Okay.
So say human chromosome 2.
Okay. That makes sense.
Yeah, so you've got chromosome 1 genotype, chromosome 2 genotype, separated by a semicolon.
Okay.
And in the example above, for example, we might just say that's chromosome 1.
Okay.
So that's all genotype related to chromosome 1.
Okay. Gotcha.
Yeah.
Okay. So to find, for this one, to find what the genotype of the parent is, do we have to find the haplotypes of this, set up a Punnett square cross, and then fill in these genotypes, and then from there find the
Right.
F1 parent?
So what I would do is, so what you're trying to do, I've given you an F1 genotype.
Okay.
So you're going to be setting up a cross. This genotype is GFP over plus, so that's one chromosome. We've got a semicolon, we've got two pluses. I'm going to use different colors to represent a different chromosome.
Yeah.
Okay. And so this is the F1 genotype.
Okay.
And it's crossed to something we're trying to figure out.
Yeah.
And that produces, so I have to work through this from scratch because it's been a while since I thought about this question. So it says you observe a 1-to-1 ratio. So it's going to be a Punnett square that has, if it's a 1-to-1 ratio of particular genotypes, then it's going to be two squares.
Okay. Okay.
And the genotypes, which is what goes in the Punnett square, are GFP over plus over plus, and plus over plus over plus.
How did you know it was only two squares?
Because of the 1-to-1 ratio. So,
Okay.
So this is, if you only get two genotypes out of a cross, then there are going to be two squares in the Punnett square.
Okay.
This is a great question. Yeah. And this F1 parent that you already know about is going to be one of the parents.
Okay.
So what are the haplotypes
Of GFP.
that it produces? You're going to have GFP with
plus. Okay. And then
and
plus with plus.
plus with plus.
Got it. Okay.
So those have to be.
Now this does not have to be on
Oh,
separate.
sorry. You're absolutely right. It's not a slash. It's a semicolon.
Gotcha.
Very good.
Okay.
Yep. So GFP over plus is that haplotype.
Okay.
And plus over plus contributes that.
Okay.
So from there, what did the haplotypes produced by the unknown parent have to be? So there's only one. So in other words, what goes there on the Punnett square?
Is it just plus plus?
Yeah. That's what's missing, right? That was what's unaccounted for in these squares. So the haplotype of that parent had to be plus plus. So what sort of a genotype do you have to have that only produces plus plus, one type of gamete, plus and plus?
Homozygous.
Yeah, it's got to be plus over plus, plus over plus.
Okay.
That's the only genotype that can make only gametes that have that haplotype.
Okay.
If you had had anything else, you would have seen other types of haplotypes here, and you would have had more rows in the Punnett square, and you would have seen more different types of genotypes in the F2.
So what's
generation.
an easy way to find the haplotypes? Like, I know that you can do the long way where you you would double this, and double this,
Right.
and then do the independent assortment.
Yeah, that's what I do when I. So I think it may be the long way, but that's the way I think about it. So it's basically a Punnett square. So the plus over plus can only give pluses.
Okay.
Although you could write it like a Punnett square if you really wanted to. You could say you either get that plus or you get that plus in a gamete.
Okay.
And you either get GFP or you get plus from this other chromosome.
Okay.
So what are the possible combinations you produce? It's
plus GFP.
Yeah, it's either GFP in both of those boxes
Okay.
or plus, and then all of these get pluses from the second gene.
Okay.
And that's exactly where you get 50% of the gametes are GFP semicolon plus, and the other 50% are plus and plus.
Okay. That makes sense. So
you have. So question 2 from the study guide. If you've got a big T, oh, good, I'm using red. If you have a big T and a little t, then after DNA synthesis you get replication, then you go through the first meiotic division, and the big T's stay together, and the little t's stay together, and you go through the second meiotic division, produce 4 cells, like that. So over this question number 2, draw a representation of all of the gametes that this individual produces over its lifetime. This works. You've got 50% big T and 50% little t. So just drawing out meiosis is a graphical representation of the frequency, the relative frequencies, how many big T's versus little t's are going to be produced. And the fact that in a monohybrid, big T over little t, meiosis always works exactly the same way. You get exactly 50% big T and little t, because you've got 4 molecules of DNA, you've got 2 divisions.
Yeah.
So it's always going to be 2 big T gametes and 2 little t gametes. So you could also diagram that as a Punnett square. Well, you could. You don't have to. I've also seen students diagram it as a column chart. So frequency, you've got big T, little t, half, 0.5. So every time a heterozygous cell goes through meiosis, absolutely guarantee you it's going to be 50-50. Every meiosis. If you add up the hundreds or thousands of millions of meioses that happen over an individual's lifespan, no change. Because every time meiosis happens, it's 50-50.
Yeah.
Do that a million times?
It's the same thing.
Same thing, exactly 50-50.
Oh, okay. I
Yep.
misinterpret the question to how many different haplotypes you would have. Like,
Oh, how
like
many total?
crosses.
Yeah, right.
And I was thinking,
Well,
wouldn't it just depend?
Totally. So I totally understand where you're coming from. Yeah, if I wanted you to just tell me how many gametes are produced over an individual's lifetime, good grief, what sort of information would I have to tell you? I'd have to tell you what the length of the lifetime was and how frequently meiosis happens. This is more like a physics question. So if an organism lives 50 years and it undergoes meiosis every month, then you could answer that question.
Yeah.
As long as I told you what age it reached sexual maturity. Yeah. So sorry. It was, I meant it to be a simple question, but it was so simple that it led to overthinking. Yeah.